Hey everybody, welcome back. Today in our series, Life Sciences Made Easy, we are looking at DNA code of life, specifically the structure of nucleic acids and DNA. And this is going to help our grade 12 learners pass that exam. So we have two types of nucleic acid. We have deoxyribonucleic acid, which is our DNA, and we have ribonucleic acid, which is our RNA. And how we get these names or these abbreviations is DNA, DNA, and RNA. Now we're going to look at or recap the nucleus of the cell because our DNA is found in the nucleus and one of the RNAs are made in the nucleus. So our nucleus is made up of four basic parts. We have the nuclear membrane, which is double and continuous with the endoplasmic reticulum. Also, the nuclear membrane contains pores in its wall, which means it basically has these little holes in the wall that allow substances to move out. It contains the nucleoplasm, which is essentially nuclear sap, and this contains our free nucleotide bases. And all of this thing will tie in as we go through this lesson. And the third structure is the nucleolus. And this structure manufactures and contains ribosomal RNA. Our chromatin network is the structure that contains DNA, and when this unwinds, it forms chromosomes. And this should somewhat be familiar because this is just recapping our grade 10 syllabus. So when we look at the structure of our nucleus, remember the nucleus is found inside the cell. You have the nucleopore, which is the structure here. This spaghetti-looking structure right here is the chromatin network or chromatin material. Some papers have either one, but it is the same thing. This structure here in the middle, or this round structure here, is the nucleolus. This in between structure is the nucleoplasm, and this double membrane structure is the nuclear membrane. So this is just a recap of the nucleus. Again, this was taught in detail in grade 10 because it's the basis of our cell and the basis of fundamentals of our cell. So let's just do a quick application questions on what we just did. When we look at this, here you have the nucleus, and here one of the statements that are made that is continuous with the endoplasmic reticulum. So this here is the endoplasmic reticulum. So first question says, letter and name of the part for each of the following. The part that unwinds to form or become chromosomes. So the part that becomes the chromosomes is my chromatin network, which is this, which would be D. The site of protein synthesis. Now, protein synthesis occurs at the site of the ribosomes, and the ribosomes would be F, which is these structures here on the endoplasmic reticulum. The part in which DNA will be found, again, DNA is found in the chromatin material. So again, it's D chromatin material or chromatin network. What does A, B, and C make up? It makes up the nucleus. And what is the nucleus' two main functions? It contains hereditary information, that it means characteristics that is passed from parent to offspring. It also controls protein synthesis, meaning the manufacturing of protein within the cell. And it also controls the functioning of the cell. So that is just 3 functions that it does have. And now here, we're not going to actually draw it, but if we look at it, it says, make an outline drawing of a plant or animal cell, which we're not going to do, but it says, add to your drawing the organelle that is referred to as the powerhouse of the cell. So this is just, again, recapping grade 10 stuff. What is the powerhouse of our cell? The power of our cell is the mitochondria. Why? Because the mitochondria produces ATP, which is energy. So going back to our content for grade 12 is now we're looking at the structure of the nucleic acid. My nucleic acid is made up of a number of smaller units called monomers, or monomers called nucleotides. So the same way our tissues are made up of cells, or organisms are made up of cells, which is the smaller units, our nucleic acids are made up of nucleotides. Now, what is a nucleotide made up of? A nucleotide is made up of a nitrogenous base, a sugar portion, so depending if it is DNA or RNA, the sugar portion would be different. Remember, DNA and RNA are examples of nucleic acid. So depending on whether it's DNA or RNA, the sugar portion would be different, and it will contain a phosphate ion. So this is a basic structure of a nucleotide. Here it says pentose sugar, which is our sugar portion. This is our nitrogenous base, and here is the phosphate ion. Now, when we look at the structure of the cell and where the DNA is, so this is the cell, all right? This would be my nucleus. So that's my nucleus. Eukaryotic cell meaning a cell that contains a nucleus. Now we zoom in on one of the chromosomes. So that's the chromosome here, and we're zooming in. And when we look at the makeup of the chromosome and we unwind it, we see that we have proteins called histones that our DNA is wrapping around. Now it's double helix because there's two strands and they are twisted. And our nitrogenous bases, or our base pairs, hold the two strands together. So this, when you look at it, it's just zooming in to exactly where the DNA is. How come we have these two strands, and what holds the two strands together? We're going to now delve into looking at the actual structure of DNA. So remember, nucleotides are going to make up my DNA because DNA is a nucleic acid and nucleotides make up nucleic acids. Because it's DNA, the sugar portion of my nucleic acid would be deoxyribose. We will have nitrogenous bases and we're going to have the phosphate ions. Now, the nitrogenous bases are divided into two groups, the purines and the pyrimidines. So here where we have nitrogenous bases, it's divided into purines and it's divided into pyrimidines. There are four types of nitrogenous bases. We have adenine, guanine, cytosine, and thymine. So once again, my nitrogenous base is divided into purine and pyrimidine. Those are the two groups. And the four types of nitrogenous bases that I actually have is adenine, guanine, cytosine, and thymine. So how do you remember which one goes for which? So just a quick tip is pyrimidines have the letter Y in it, and cytosine and thymine have the letter Y in it as well. So these two will fall under the group pyrimidines. Because these two fall here, guanine and adenine will fall under purine. So just that's a quick tip. Now my sugar portion, or my sugar molecule of one, so this is how we are actually creating this long chain of our DNA. The sugar molecule of one nucleotide, so this is my sugar molecule of one nucleotide, attaches to the phosphate ion of another nucleotide. So my sugar portion here is going to attach to my phosphate ion here. And my sugar portion here is going to attach to my phosphate ion here. And it attached by these bonds called sugar phosphate bonds. So this is now creating my chain. And remember, this is only forming one strand. Another chain has to attach on this side, because my nitrogenous base is going to pair up with the other nitrogenous base over here to form the two strands. Cytosine only pairs with guanine, and adenine only pairs with thymine. And these are called complementary base pairs. This means that guanine and adenine will never pair together, or cytosine and adenine are never going to pair together. They will pair with their complementary partner. So CG will always go together and AT will always go together. And now how these two nitrogenous bases actually hold or pair together is via a weak hydrogen bond. Now, quick tip before we go further. This word weak is very important. If you leave out the word weak, many times you will get the answer marked wrong. So these weak hydrogen bonds hold the nitrogenous bases together, and this is going to form our double-stranded structure. So here you see a strand here, a strand here. My adenine from here is joining or bonding to my thymine on this side. My cytosine up here is bonding with my guanine on the other side. And here I have my sugar phosphate portion, sugar phosphate bonds. So this is one strand and this is another strand. As a result, it's going to end up being a ladder-like molecule that twists so that one coils around the other, that forms the double helix structure, the one that's twisted. Now, the functions of DNA. It controls the synthesis, which is the manufacture of proteins, and it transmits hereditary characteristics from parent to offspring. Now, this one here controls the synthesis of proteins. Now, specifically looking at the synthesis of protein, the fact that DNA controls the synthesis of proteins, there are multiple in which it actually controls. So, proteins that are synthesized by cells, these are multiple. Muscle, connective tissue, which is our collagen, bone tissue, keratin, legumin, seeds, all of these are proteins. Enzymes are a form of proteins, and they are specialized because they control all metabolic activities within the cell. So practically any reaction that is occurring in our body or in our cells, enzymes are controlling them. It controls the structure and functioning of the cells, so it controls the organism as a whole by controlling the manufacture of proteins. And in chloroplast and mitochondria, it controls the protein synthesis in these organelles. So in chloroplastic DNA and mitochondrial DNA, it will control protein manufacturing or synthesis in the chloroplast and the mitochondria. With regard to transmits hereditary characteristics from parent to offspring, it basically ensures that DNA is being passed on from the existing cells to new cells during reproduction. So let's look at, again, breakdown to where we are now. So our nucleotide is made up of a sugar portion, a nitrogenous base, and a phosphate ion. My phosphate, this is a very simplistic part of it, but my phosphate, my sugar over here will join to a phosphate ion of the next nucleotide via a sugar phosphate bond, and that's why it says over here sugar phosphate backbones. The nitrogenous base can be either adenine, thymine, cytosine, and guanine. Now, if you look at this diagram, you can see that all the adenines and the thymines are paired or bonded. And all the cytosines and guanines are all paired or bonded. Now, because these bonds are weak, it causes uh, the ladder structure to now wrap around each other to form this double helix structure that we see here. Helix means to be twisted. So it is a twisted ladder. So our exam tip that I have for you all, once again, pyrimidines are made up of cytosine and thymine because cytosine and thymine have the letter Y in it. The sugar of the nucleotide indicates whether it's DNA or RNA. For example, deoxyribose for DNA. You must include the word weak when indicating hydrogen bonds. If you leave out that word weak, you are going to get your terminology wrong and you're going to lose marks or you will not be credited for that. Your adenine always pairs with adenine always pairs with thymine. And how I remembered it by just abbreviating it as AT. So I always know adenine is pairing with thymine, and as a result, guanine is pairing with cytosine. You can find a acronym that helps you all remember. These are just my tips to help you all remember. And once again, learn your diagrams in black and white. Learn it in color, understand it, but then go over it again in black and white. Because in that exam, when you're sitting to write it, it's not going to be in color. It's going to be in black and white. So you must be able to identify the different structures when it is in black and white. So now let's look at an application question. So when we get a diagram like this, make sure you read all the relevant information and look at the diagram carefully to see what is going on. If I look at it, it says that it represents some nucleotides in a single strand. So it's not showing us the entire DNA structure. It's just showing us one strand of the DNA structure. And it's showing us that it's reading from top to bottom. This would be my sugar portions. This would be my phosphate ions, and these are my nitrogenous bases, which has made it easier because they've actually indicated the nitrogenous bases on it. Then it says, give the letter of the part that represents a sugar portion or the sugar molecule. So it's going to be Y. Phosphate molecule, it's going to be X. How many nucleotides are represented in the diagram? So remember, your nucleotide is made up of a sugar, a phosphate, and a nitrogenous base. So that will be 1, 2, 3, and 4. So there's 4. Then they say, write down the nitrogenous bases of the complementary DNA strand, and it says specifically from top to bottom. So on the complementary strand that should be here, what would be the nitrogenous bases for each one from top to bottom? So thymine is going to attach to adenine, guanine, cytosine, adenine, thymine, and cytosine, guanine. So if I'm just giving, that's not answering the question. The question is though, to just write the nitrogenous bases of the complementary strand. So the answer should be A, C, T, and G. Then they say, for the next question, name 2 processes that require the 2 strands of the DNA molecule to separate into single strands as we see it here, which we will be covering in future lessons. But the 2 processes would be protein synthesis and it will be DNA replication. So until next time, stay curious and keep learning.